Here's the big idea for this talk. Bacterial infections don't just happen. They follow a logical sequence of steps. And if you understand the steps, you can understand why certain bacteria cause certain diseases, why some animals are more vulnerable, and where we can intervene. We're not going to memorize a list of pathogens. We're going to build a mental model of pathogenesis that you can apply to any bacterium you encounter. I want you to hold on to this six-step model like a framework. Every time a patient comes in with a bacterial infection, you're essentially asking, where did this pathogen succeed that it shouldn't have? Did the animal aspirate a huge dose? Did the immune system fail? Was there a break in the mucosa? This isn't just academic. It tells you how to treat and it tells you how to prevent. You block any one of these steps and the infection doesn't happen. Vaccines work on step 1 and 3. Antiseptic technique works on step 1. Antibiotics work on step 4. Here's something I find fascinating about pathogen biology. Bacteria don't just land anywhere. They have molecular Velcro, pili and adhesins. And the host cell has matching receptors. E. coli strains that cause UTIs have pili that bind specifically to uroepithelial cells. Bordetella bronchoseptica has adhesins that bind ciliated respiratory epithelium. This is why the same species of E. coli can cause diarrhea in one context and a kidney infection in another. Different strains carry different adhesins targeting different tissues. The lock and key match between adhesin and receptor determines where the infection happens. The host immune system is genuinely remarkable at clearing bacteria, so pathogens have evolved a dazzling array of tricks to survive it. Some hide inside the very cells sent to kill them. Brucella and Mycobacterium are masters of this. They get swallowed by macrophages, then prevent the phagosome from fusing with the lysosome. The macrophage cannot digest them. They're living inside their would-be killer. That's why intracellular pathogens are so hard to treat. Most antibiotics don't penetrate macrophages well, and that's why Brucella treatment requires long courses of 2 drugs. The biology drives the therapy. This is one of the most important conceptual shifts in infectious disease. The host's own immune response is frequently a major contributor to the pathology we see clinically. Think about a severe pneumonia. Yes, the bacteria are damaging epithelium, but it's the massive neutrophil influx, the cytokine storm, the edema and fibrin that fills the alveoli that actually compromises gas exchange. Or think about Clostridium perfringens gas gangrene. The alpha toxin is destroying cell membranes and cutting off blood supply faster than the immune system can respond. Some organisms are vicious direct killers. Others are puppet masters that make the immune system do their dirty work. Exotoxins are some of the most potent biological molecules known. Botulinum toxin, produced by Clostridium botulinum, is the most toxic substance known by weight. It cleaves the snare proteins that allow acetylcholine vesicles to fuse with the presynaptic membrane. No fusion, no neurotransmitter release, no muscle contraction, flaccid paralysis. Tetanus toxin does the opposite. It blocks inhibitory interneurons, causing spastic paralysis. 2 toxins from the same genus doing opposite things to the nervous system. Once you understand the mechanism, the clinical signs make complete sense. We've spent a lot of time on what bacteria do, but here's the other side. The host matters just as much. Think about a feedlot. You can have the same Mannheimia hemolytica exposure in 100 calves and maybe 15 get pneumonia. What's different? Stress from transport suppresses neutrophil function. Weaning stress elevates cortisol. Concurrent BVD virus strips immune cells. A concurrent viral infection may have already damaged the respiratory epithelium. The bacterium didn't change, the host susceptibility did. This is why stress management and vaccination protocols are legitimate infection control tools. What I want you to carry out of this talk. Pathogenesis isn't a list of facts about specific bacteria. It's a framework. The 6-step sequence applies to every bacterial infection you will ever encounter. When you're standing at a sick animal, ask yourself, where did this organism succeed? Where did the host's defenses fail? That mental model will guide your diagnosis, your treatment decisions, and your prevention advice. It's not about memorizing, it's about reasoning. The biology is logical. Follow the logic. Read case question 1, give it a thought, and we'll work through it together. This is a perfect exotoxin question. See, tetani is not invasive. It stays at the wound site, but it produces tetanospasmin, a neurotoxin that is absorbed and travels retrogradely up motor neurons to the spinal cord and brainstem. There, it cleaves synaptobrevin, blocking the release of inhibitory neurotransmitters, glycine and GABA, from Renshaw cells. Without inhibitory input, motor neurons fire unchecked. The result is sustained muscle spasm and rigidity, the sawhorse stance, trismus, hypersensitivity. Death comes from respiratory muscle failure or aspiration. The bacteria never had to go anywhere. The toxin did all the work. That's the power and the danger of exotoxins. Think about the question. We'll then work through the answer together. Multiple host factors converge. First, neonates are hypogammaglobulinemic at birth. They have essentially no circulating antibodies until they absorb colostrum. If colostral transfer fails or is inadequate, they're immunologically naked. Second, their complement system is immature. Third, their neutrophils don't migrate as efficiently. Fourth, the integument and mucosal barriers, especially the umbilicus and gut, are still immature and easily breached. Fifth, they haven't developed immunological memory. So you have an underdeveloped, underarmed immune system meeting a pathogen for the first time, often in a high exposure environment. The result is overwhelming sepsis from bacteria that a healthy adult would clear in hours. Failure of passive transfer is the single most important risk factor for neonatal sepsis in large animals. About this case, and we'll work through it together. The normal gut microbiome is a critical defense mechanism. It competes with pathogens for nutrients and attachment sites, produces short-chain fatty acids that acidify the environment, stimulates local immunity, and physically occupies niche space. When you disrupt it with broad-spectrum antibiotics, a sudden diet change, or stress, you create openings. Opportunistic pathogens like certain Clostridium species or E. coli strains that were present at low numbers suddenly have no competition. They bloom. This is called dysbiosis associated disease, and it's directly analogous to C. difficile colitis in people. The pathogen was often already present. The host ecosystem just stopped holding it in check.